Welcome, curious minds, to the deep dive. Today, we're pulling back the curtain on one of life's most intricate and fundamental processes, meiosis.
Mm-hmm.
And if you're one of Mr. Chi's dedicated students, or maybe just someone who loves unraveling biology mysteries, you're definitely in the right place.
Absolutely.
Our mission today is to take this, complex cellular choreography and make it crystal clear for everyone, really, whether you're just starting out or, eager for all the details.
Indeed. And meiosis isn't just some biological detail you memorize. It's really a core engine of diversity. It's a cornerstone for understanding how species reproduce and, adapt.
Right.
Grasping this process, it unlocks so much more in biology.
Absolutely. So we're going to extract the most important insights from our sources today, try to demystify the complexity of meiosis.
Sounds good.
We'll look at why it's so crucial, walk through the step-by-step process, the actual dance moves, if you will, compare it to its cousin, mitosis, and even touch upon what happens when things go a little bit wrong. Okay, let's unpack this then. We often start with mitosis, right? Which you might remember as the body's kind of general cell division process. Think of it as the maintenance crew.
Exactly.
It's how cells divide for growth, for repair, making exact copies of our somatic cells, our body cells.
Right.
So if you get a cut, mitosis is working there to heal the wound.
Yeah, making identical cells.
But meiosis, it's a completely different kind of event, a truly special type of cell division. And its core function is basically to have the number of chromosomes.
And what's truly fascinating here isn't just what it does, the having, but why this is so incredibly critical. Meiosis is, indispensable for life as we know it for three main reasons.
Okay.
First, it's all about producing haploid gametes.
Haploid gametes.
Yeah, these are your specialized sex cells, sperm and egg. And crucially, they each contain only half the number of chromosomes. So if a regular body cell has the full set, a gamete just has a half set.
Okay, so instead of the full instruction manual, they get just one copy of each chapter.
Yeah.
That makes sense if two are going to combine later, but why is that so essential? Why not just combine two full sets?
Well, that leads right to the second reason. Overcoming the doubling effect of fertilization.
Ah, right.
Imagine if gametes didn't have their chromosome count, with each new generation, the number would just double.
So 46 chromosomes becomes 92, then 184.
Exactly. It would get out of hand really fast. That's simply not sustainable for a species.
Okay.
So meiosis ensures that when two of these haploid gametes fuse during fertilization, the original constant chromosome number for that species, like 46 for humans, is perfectly restored. It's a really beautiful balance.
And that balance, that constant number, it leads us to the third reason, right? Which I think is maybe the most exciting part.
I think so. Meiosis is absolutely essential for generating genetic variation.
The differences between us.
Precisely. This is the raw material for evolution, for adaptation. Thanks to the unique things that happen inside meiosis, the offspring produced are genetically unique, not just from their parents, but often from their siblings too.
Wow.
This incredible diversity is what allows populations to adapt to changing environments. It drives natural selection. I mean, think about it. Without this cellular reshuffling, life would be far less resilient, less adaptable.
Right. To really get this process, though, we need to make sure we're all speaking the same language. New topics always come with new terms.
Vocabulary is key.
Exactly. So mastering the terminology is step one, especially for students just grappling with this.
Yep.
We'll introduce key players as we go, but let's start basic. When we talk genetics, the first word is usually chromosome.
Right. A chromosome is basically a thread-like structure. You find it in the nucleus of most living cells. It's essentially a tightly packed bundle of DNA and protein, and it carries the genetic information of the genes.
Like a chapter in the instruction manual.
That's a good way to think of it. Yeah, a specific chapter for building and operating an organism.
Okay. And then, after DNA copies itself, these chromosomes look a bit different when you start talking about chromatids.
Precisely. So a chromatid is one of the two identical strands of a replicated chromosome. If you picture that replicated chromosome looking like an X, each arm of that X is a chromatid. They're identical copies made during interphase, and they're held together at a central point.
And that central point holding them together.
That's the centromere. It's like the constricted region, the waist of the X that holds the two sister chromatids together.
Got it. We also hear about homologous chromosomes. What makes them homologous?
Okay. Homologous chromosomes refers to a pair of chromosomes. You get one from each parent. They're the same shape, same size, and they have similar genes in the same locations or loci.
So they carry genes for the same traits, but maybe different versions, like eye color.
Exactly. They're a matching set in terms of the genes they carry, but they aren't identical copies because one came from mom and one from dad.
Okay. And the terms we mentioned earlier, diploid, 2N, and haploid N. Let's pin those down.
Right. Diploid, or 2N, means a cell has 2 complete sets of chromosomes, 1 set from each parent. That's like most of our body cells.
Our somatic cells?
And haploid, or N, means a cell has only 1 complete set of chromosomes. That's exactly what we see in the gametes, the sperm and egg cells.
So diploid is the before meiosis state for the parent cell, and haploid is the after meiosis state for the gametes.
You got it. Those terms describe the chromosome count. And one more quick one, interphase. That's the phase before meiosis even starts, where the crucial DNA replication happens. That's when single chromosomes become the double-stranded X shape, forming those 2 sister chromatids.
Okay, good ground covered. All right, let's get to the main event. The meiotic dance itself. Whoa. Meiosis isn't just one division, it's 2 acts, meiosis the 6th and meiosis the 2nd.
2 major divisions.
And just to make it easier to follow, let's imagine we're watching a cell with only, say, 4 chromosomes total, instead of the human 46. The principles are identical, just easier to track.
Good idea. And it's crucial to remember that meiosis the 1st is often called the reduction division.
Reduction. Because that's where the chromosome number gets cut in half.
Exactly. That's the key event of meiosis the 1st. Meiosis the 2nd, then, is more about separating the sister chromatids. It actually looks a lot like mitosis in many ways.
Okay. And for both divisions, we use the same 4 phase names. Prophase, metaphase, anaphase, and telophase. That PM8 mnemonic helps.
PM8T, yep. Helps keep track.
So, meiosis the 1st, the reduction division.
First up, prophase I. This is where the stage gets set, and like you said, it gets really interesting. The nuclear membrane, the nucleolus, they start to disappear, clearing the stage. The centrosomes split, they form the spindle fibers, those ropes and pulleys, and the chromatin, which is usually all loose, it condenses into those visible, distinct chromosomes.
And here's the absolute key event, unique to prophase I. The homologous chromosomes pair up. They lie side by side in perfect alignment.
The ones from mom and dad find each other.
Precisely. This pairing forms what we call a bivalent. And within this bivalent, something amazing happens. Crossing over.
Ah, the genetic shuffling.
Yes. The inner chromatids from each homologous chromosome actually overlap. They touch at specific points called chiasmata.
Chiasmata, okay.
And at these points, segments of the chromatids literally break off and swap places between the homologous chromosomes.
Whoa.
This exchange of genetic material is profound. It's a major source of genetic variation. It ensures the resulting gametes are truly unique combinations of the parental genes. It's like shuffling the genetic deck right at the start.
Talk about a genetic remix. Okay, next up is metaphase I. What happens here?
In metaphase I, these homologous chromosome pairs, the bivalents, they all move to the middle of the cell. They line up along the cell's equator.
The pairs line up together.
Yes, the pairs. And each chromosome in the pair attaches to a spindle fiber from opposite poles of the cell.
And is there randomness here too?
Absolutely.
Yeah.
This brings us to another critical source of variation. Random arrangement or independent assortment. The way each homologous pair lines up at the equator is completely random. Meaning which chromosome of the pair, the maternal or paternal one, faces which pole of the cell is pure chance for every pair.
Ah, so if you have multiple pairs, the combination of which ones go which way is huge.
Exactly. With 23 pairs in humans, the number of possible combinations is enormous. This independent assortment further shuffles the genetic deck, increasing diversity.
Okay, so after they line up randomly, anaphase I.
Anaphase I. Yeah. This is a critical separation. The whole homologous chromosomes are pulled apart. They move to opposite poles, guided by those contracting spindle fibers.
The whole X shape moves.
Yes, the entire replicated chromosome, still with its 2 chromatids joined at the centromere, moves. The crucial point here is the centromeres do not split yet.
Okay, so the pairs separate, but the sister chromatids stay together.
Precisely. Homologous chromosomes separate.
Got it. And that brings us to the end of meiosis the 3rd, telophase I.
Right. In telophase I, new nuclear membranes start to form around the groups of chromosomes now clustered at each pole. The nucleolus might reappear.
And then the cell itself divides.
Yes. Following telophase I comes cytokinesis, the division of the cytoplasm. This splits the original cell into 2 distinct daughter cells. And here's the vital outcome of meiosis 3. These 2 cells are now haploid.
Because the homologous pairs separated?
Correct. Their chromosome number has been halved. And importantly, they are genetically different from each other and from the original parent cell, thanks to crossing over in that random arrangement.
Wow. Okay, so 1 division down, 2 unique haploid cells created.
Now for meiosis the 2nd. separating the sister chromatids.
So meiosis the second, this happens in both of those cells, which is made in meiosis the second.
Correct. Both daughter cells from meiosis immediately or after a short pause enter meiosis the second. And as we mentioned, it looks quite similar to mitosis.
Okay, so prophase 2, what's happening?
In prophase 2, if nuclear membranes reformed in telophase to first, they disappear again. Same with the nucleolus. The spindle apparatus forms again in each cell. The key thing is, the chromosomes here are not in homologous pairs anymore. Each chromosome still consists of 2 sister chromatids, but they're unpaired.
Right, because the pairs split in anaphase the first, so metaphase 2.
In metaphase 2, the individual chromosomes, each still an X shape, with 2 chromatids line up along the equator of each of the 2 cells.
Single file this time, not in pair.
Exactly. Single file, randomly arranged along the equator. Each chromosome attaches to spindle fibers from opposite poles.
Okay, and then anaphase the second. This must be where the X splits.
This is the moment. In anaphase the second, the centromere finally splits.
Yeah.
And with that split, the 2 sister chromatids are pulled apart. They move to opposite poles of the cell. And now each of those former chromatids is considered an individual unreplicated chromosome.
So the sister is separate.
Sister chromatids separate, yes.
And that leads us to the grand finale, telophase 2.
Telophase 2 wraps everything up. New nuclear membranes form around the sets of now unreplicated chromosomes at each pole in both dividing cells. The chromosomes begin to decondense.
And then cytokinesis again.
And then cytokinesis happens again, dividing the cytoplasm of each of the 2 cells that went through meiosis II. So the final result, a grand total of 4 daughter cells.
4 cells from the original one.
Yes. And each of these 4 cells is haploid. And crucially, they are genetically different from each other and, of course, from the original diploid parent cell. They carry those unique genetic combinations forged during both meiosis and 3 and 2. It's really an intricate ballet of division and recombination.
So, okay, putting it all together, comparing mitosis and meiosis feels really important now. We've seen the steps, but what are the big picture differences? If mitosis is the copier,
then meiosis is the generator of diversity. Let's nail down those key distinctions. It really helps clarify things.
Okay, first up, where it occurs.
Right. Mitosis happens in your regular body cells, your somatic cells, for growth, repair, general maintenance. Meiosis, though, is restricted. It only happens in the sex organs, tests in ovaries in animals, anthers and ovules in plants, specifically to produce gametes.
Big difference than the number of divisions.
Clear cut. Mitosis involves just 1 round of division. Meiosis takes 2 full rounds, meiosis the first and meiosis the second.
Which leads directly to the number of daughter cells.
Yeah. Mitosis gives you 2 daughter cells. Meiosis gives you 4.
And probably most important difference, genetic identity.
Absolutely key. Mitosis produces daughter cells that are genetically identical clones of each other and the parent cell. Meiosis, it produces daughter cells that are genetically different from the parent and from each other, thanks to crossing over and random assortment.
Okay, and the chromosome number.
Mitosis maintains the chromosome number. Diploid parent cell, 2N, makes diploid daughter cells, 2N. Meiosis halves the chromosome number. Diploid parent cell, 2N, makes haploid daughter cells, N.
Makes sense for gametes. And finally, that really cool event, crossing over.
Crossing over, that vital source of variation, happens only in meiosis, specifically during prophase of 1. It does not happen in mitosis.
So lots of clear differences.
Definitely. Although it is worth pointing out the one main similarity.
Oh, yeah.
The actual mechanics of meiosis II, how the chromosomes line up in metaphase 2, how the centromeres split and chromatids separate in anaphase 2, that process is very, very similar to what happens in mitosis.
Ah, okay. So meiosis II is kind of like a mitotic division, but happening in cells that are already haploid.
That's a great way to think about it. It helps connect the 2 processes in your mind.
Now, we've talked about this intricate, beautiful dance, but like any complex process, I guess sometimes mistakes can happen.
That's right. While meiosis is usually incredibly precise, sometimes missteps occur. The choreography isn't perfect every single time.
And what kind of mistakes are we talking about?
Well, the most common type of error is something called non-disjunction.
Non-disjunction, okay.
It literally means not coming apart. This happens when either the homologous chromosome pairs fail to separate properly during anaphase I.
So a whole pair goes one way.
Exactly. Or it can happen later when sister chromatids fail to separate correctly during anaphase II.
So one pole gets both sisters, the other gets none.
Right. Either way, it's an error in distributing the chromosomes. The end result is that the gametes produced can end up with an abnormal number of chromosomes, either too many or too few.
And what are the consequences when that precision goes wrong? When gametes have the wrong number?
Well, a very well-known and impactful example is Down syndrome.
Okay.
This condition is usually caused by non-disjunction involving chromosome pair 21.
Chromosome 21 specifically.
Yes. If non-disjunction happens with chromosome 21 during meiosis, usually in the egg, it results in an abnormal gamete, an egg or sperm, that contains an extra copy of chromosome 21.
So it has 24 chromosomes instead of 23.
Correct. And when this abnormal gamete fuses with a normal gamete during fertilization, the resulting zygote will have 3 copies of chromosome 21 instead of the usual 2. That gives a total of 47 chromosomes instead of 46. This specific condition, having 3 copies of a chromosome, is called trisomy 21.
Trisomy 21. And what are some of the characteristics associated with that?
Individuals with Down syndrome often share certain physical characteristics like upwardly slanted eyes, maybe a small nose with a flattened bridge, a smaller mouth. There's also typically some degree of intellectual disability, which can vary quite a bit.
Okay.
Other common features might include decreased muscle tone, sometimes hearing loss, or even heart defects present at birth. It results in a range of developmental differences.
Is there anything known to influence the chances of non-disjunction like this happening?
Yes, research clearly shows that the chance of non-disjunction occurring generally increases with parental age, and particularly with the mother's age as our eggs get older. And because of this, for older parents, or if there's a family history, diagnostic tools like amniocentesis are available during pregnancy.
How does that work?
Amniocentesis involves carefully taking a small sample of the amniotic fluid surrounding the fetus. This fluid contains fetal cells. These cells can then be used to create a karyotype.
A karyotype? That's like a picture of all the chromosomes.
Exactly. It's a visual map, arranging all the chromosome pairs by size and shape. This allows medical professionals to count them and visually inspect them, clearly detecting chromosomal abnormalities like trisomy 21. It gives parents really important information.
Wow, what an incredible journey we've taken. From the reasons why meiosis happens through all those intricate steps, the comparisons, and even when things go wrong, we've really unpacked the world of meiosis.
We certainly have.
Right.
We've seen its fundamental importance for reproduction, obviously, but maybe even more profoundly for generating that incredible genetic diversity that fuels life itself.
Yeah, we've reinforced how it halves the chromosomes, how it creates these genetically unique gametes, and the huge role of things like crossing over and random arrangement and driving variation.
Mm-hmm. It's the engine of individuality, really.
It's truly mind-blowing when you stop and think about it. So maybe here's a final thought for you, our listeners, to ponder. How does this incredibly detailed choreography of meiosis, with its precise steps, its moments of genetic swapping, and even its potential for mistakes, how does that highlight the delicate balance, but also the amazing resilience of life and inheritance?
That's a great point. Every single organism you see, including yourself, is a product of this cellular dance happening generation after generation.
Yeah.
It's the process that allows species to adapt and survive over vast stretches of time, facing all sorts of environmental changes. It underscores how precious and complex our genetic blueprint is.
And how interconnected life is through this shared fundamental process.
Absolutely.
Keep exploring, everyone. Keep asking questions and never stop being curious learners.
Hear, hear.
And hey, for more amazing content, especially if you're one of Mr. Chi's students or followers, make sure you follow and subscribe to Mr. Chi on TikTok, Instagram, and YouTube. Seriously, he's got fantastic resources that can help reinforce everything we talked about today, and so much more.
Great recommendation.
Thank you so much for joining us on this deep dive today and for being such engaged listeners. Until next time.